My name is Neil Watson. I'm the Chair of Psychology at SFU, and welcome to the inaugural Lollo lecture. It's a great pleasure to welcome you. I've been looking forward to this for a long time. This is an event that we've established in order to bring an eminent scientist to SFU each spring and to share that person with our friends and colleagues in the Vancouver area by staging a public lecture here at the downtown campus that everybody can get to. It's a great pleasure to have Vince. It was he makes an excellent, very photogenic poster that our good friends at Creative Services were able to put together, and I'm pleased to be able to present you with the very first one of these, Vince. I'm doubly grateful to you for allowing us to trade on your good name in two ways. The first of those, of course, is by lending your name to this lecture series into the future, but additionally by agreeing to be the first recipient of the Lollo Distinguished Lectureship in Psychology. In a moment, I'm going to turn the mic over to my colleague Tom Spalek, who works closely with Vince and will introduce him properly. But I just wanted to say that there'll be ample opportunity for some questions after the talk. In order to do that, if you would put up your hand, I'll run over with a mic so that we can capture it on tape. We're being recorded today. And following that, we're going to have a reception nearby with refreshments, and everybody is most welcome to join us for that. So, Tom, I'll turn it over to you.
So, back about 85 years ago in October of 1930, something big happened in Europe. Turkey and Greece, after centuries of war, signed a peace agreement. And I think the momentous sort of nature of that overshadowed something else that happened in a little town in Gorizia, which is on the border of Italy and Slovenia. Because if that hadn't happened, I'm sure the papers would have all been running stories such as this. And you know that the first thought that came to Vince's mind when he came out would have been something like this: for non-Italian people, it is all due to reentrant processes. So I'm not going to spend a lot of time on Vince's childhood. There's lots of interesting stories that Vince could obviously tell you about that, including being a black market flower runner. But what I will sort of focus in on is after that. In 1950, after the war, Vince and his family had spent several years in refugee camps. And in 1950, they boarded a boat something like this and made the trek from Italy through the Suez Canal and to Perth, Western Australia. Not being terribly familiar with the English language, Vince had to take various jobs in order to make meet financial requirements. And some of those jobs that he worked over the 5 years, one was as a barman, where he demonstrated the psychological principle of one-shot learning. Because, as Vince tells it, the locals one night plied him with alcohol and asked him to sing, and they never asked him to sing again. In addition to working as a barman, he also worked as a laborer in a steel foundry, and as a stonemason's assistant, and finally, as a grizzly man. And for those people who aren't familiar with the term grizzly man, it's a person who works in a mine, in this case a gold mine, and these iron bars here have these little sort of slots in it for the ore to fall through. And that sort of grid is called a grizzly, and the grizzly man's job is to break up the ore so it's small enough to fall through the cracks. And he could either do so with his trusty hammer, or he could also use his waterproof matches in order to blow the ore up. And Vince tells me that they don't call it gelignite, they call it fracture. So you can see that a lot of these jobs involved a lot of physicality and adrenaline, and so you can imagine that after doing this for 5 years, Vince probably looked something like this. Having achieved physical perfection, he decided that the next step was to focus on mental perfection. And so in 1935, he enrolled in, where was it? Yeah, 1955, all right. Wasn't 5 years old. He managed to secure a scholarship to the University of Western Australia and enrolled in the teacher's training program there. And so this is a photo of his class, And I challenge anybody who hasn't seen this picture before to find Vince Lollo in the crowd. There is no distinctive cue. There are no mustaches, so you can't focus on that. And this is Vince here. So during his undergraduate career, he was doing a double major in geography and psychology, and he managed, as an undergraduate, to do one of the unheard of feats of actually doing some research in geography and managed to get his work published. So in second year of undergraduate, he managed to publish a paper to resolve a controversy that was present in the literature regarding the tides at Fremantle, where they were trying to decide if the tides were daily or semi-daily. And Vince figured out that it was all due to the declination of the moon and resolved the controversy. And having figured out that he could do this as an undergraduate in second year, that obviously geography is not where the challenge is, and so if he wanted mental perfection, he needed to actually focus in on something more challenging, and so he did his honors in psychology. In 1959, he graduated with his BA and achieved the distinction of first-class honors, which is an extremely rare designation, and allowed him to go directly into the PhD program. He managed to get his PhD in an astonishing 3 years, and from that, his work was so innovative in that he was also able to secure a Fulbright scholarship to study in the States, where he studied at Indiana University, Michigan, and Princeton from 1962 to 1965. And during this time, he was what he likes to refer to as a rat runner. And so he did a lot of work using the rodents in order to figure out all the sort of stuff that he wanted to figure out. And there's some of you in the audience who are familiar with running rats and that. In 1965, having completed the Fulbright scholarship, he returned to the University of Western Australia as a lecturer. And the event was so momentous that the paper actually did run a story, sort of acknowledging his return. And so that's Vince again. And the paper also sort of acknowledged that Vince managed to secure a grant from the US Air Force to do his research to the tune of about £30,000 a year, which in 1965 was a very handsome sum, which I've been told by Vince is approximately the equivalent of about $1 million nowadays. So with that, obviously, he was able to procure the top-of-the-line equipment. And again, there's a picture of Vince, sort of looking at his subject, who's about to run through a maze. So as part of his grant from the US Air Force, he was required to fly back and forth across the ocean and make regular reports to the US Air Force in the States. And on one of these trips, I've been told the University of Western Australia sent him a telegram in 1967 to let him know that he's been promoted to senior lecturer. In 1970, after being offered the chair of psychology at Melbourne and turning it down, the University of Western Australia then promoted him to the position of reader. So at no point in this time did he actually have to apply for these promotions. He was just given these promotions. Vince took a sabbatical in 1972, and because his research had switched somewhat focus from being on rat running to being more cognitive in nature, in 1972 he decided to do his sabbatical at a small little university in Ontario that some of you may be familiar with by the name of Trent University, and worked with a researcher there by the name of Doug Lowe. And experiencing the culture and the sort of welcomeness that everybody sort of showed him, he decided that Canada was the place to be. And so from that point onwards, he was constantly on the lookout for any positions that might be available. And just sort of a quick thing, this is his lab in the University of Western Australia, and you can see there's Vince there. And so in 1975, a position opened up in Winnipeg at the University of Manitoba, and so he immediately jumped on that and flew over to Canada to start his illustrious research career here. In 1978, the University of Alberta decided to headhunt him and bring him over from U of M to the U of A, and to do so they actually doubled his salary. So Vince spent the rest of his career, prior to retirement anyways, at the University of Alberta, again with a good group of students. And during this time, he did various things such as visible persistence, temporal integration, motion perception, phosphor persistence, and various other things. And all good things, unfortunately, come to an end, and the U of A at that time had mandatory retirement, and so in 1996, having already turned 65, he was forced to retire, but he had a very sort of successful career. In the time since his postdoc, his Fulbright scholarships to when he retired, he published 86 peer-reviewed journal articles, which averages out to about 2.5 articles per year. Vince served many years on the NSERC Discovery Grants Committee, and in discussions with him, he told me that for the NSERC Grants Committee, when they decided if they were to renew your grant, they basically looked for a minimum of 1 publication per year. 2 publications per year was considered pretty stellar. So the fact that Vince managed to, for the entire duration of his career, keep to a 2.5 per year sort of clip is pretty remarkable. And as they say, when one sort of chapter ends, another chapter begins, Vince decided in 1996 that he would now start a completely new research path and decided to focus in on the concept of attention. And those who were at his talk yesterday have sort of heard about what his feelings are on the concept of attention. But he decided to pursue that by coming to a place where he didn't have to worry about icicles forming on his mustache, and joined the lab of Jim Enns at the University of British Columbia. And so for those of you who are not familiar, that's Jim back there, and there's Vince. And Shahab is actually here, but he's not in that picture. Okay. All right, so Vince worked in Jim's lab for about 9 years, and during that time, he served as the president of the Canadian Society for Brain Behavior and Cognitive Science, was elected a fellow of the Royal Society of Canada, and also was awarded from the Canadian Society for Brain Behavioral and Cognitive Science the Donald Hebb Award for a career of distinguished contribution to psychology. During that time, he also now published 31 more journal articles, which translates into an average of a 3.4 per year. Again, extremely impressive. But in 2004, we managed to steal Vince away from UBC to SFU and managed to get him to join my lab. And so here's a picture of my lab. And what you don't notice is the shackle that's attached to Vince's right foot there, because we're not letting him go under any circumstances. During his time at SFU, he's also been awarded the 2007 Richard Tees Award for leadership to the area of psychology, including, as I said, serving on the NSERC Discovery Grants Committee twice, so 2 4-year terms, and during both of those terms, he also served as the chair. He also organized various sorts of grassroots campaigns, including one where he managed to sort of get rid of president of NSERC who was not doing a good job for the area, or for any area, actually. He also, in 2010, was awarded an honorary doctorate of science from the University of Lethbridge. And in the time so far that he's been in our lab, he's managed to publish 52 more journal articles, which means that now he's publishing at a clip of about 4.7 per year. So 2.5, 3.4, 4.7, it's obvious that his career is accelerating. And NSERC has recognized this in Vince's last NSERC grant application. They not only renewed his application, they increased his funding, and they also awarded him an accelerator grant in order to kickstart his research. So, without further ado, I wanted to introduce Vince Lollo to give the inaugural Vince Lollo Distinguished Lecture entitled 50 Shades of Gray Matter, A History of What We Know About the Brain. And so Vince is a fantastic colleague and a good friend.
Well, thank you, Tom. A very entertaining and a detailed introduction. It is indeed an honor to be invited to be here today. An honor in the sense that the people who matter most in one's professional life, at least within academia, are one's peers, one's colleagues. And to be singled out by one's colleagues for any kind of honor, especially the dedication of a lecture series, is indeed humbling in a way, but certainly very rewarding. Thank you. About the brain, now let me see, how do I start this gadget here? Is it on?
You just have to advance it like on.
Oh, I see. Okay. That's a nice, juicy brain. To say it with Woody Allen, who said, the brain, the second most important organ of the body. Do I have a pointer, Tom? Yes. Can you see the pointer there? Yeah. Oh. All right. Now we're going to split this brain along the sagittal axis, and we see this. I will be talking mostly about an area of the brain which is known as the calcarine fissure, that has to do with, that's where the visual stimuli first enter the brain. Now the brain, you see, encased in that 3 pounds of matter are 100 billion neurons. Now, the population of the world is 7 billion. That's 15 times the number of neurons in there. Not only, but each of those neurons connects with other neurons. Each neuron connects to about 7,000 other neurons on average. So the number of connections amongst neurons in the brain is astronomical. It's something like 500 trillion connections in that small encased hunk of matter. Now, let's start from the beginning. I said that I was going to talk about the history of what we know about the brain. Very early on, 1 million years ago, the Australopithecines in the southern part of Africa knew about the importance of the brain, obviously because, or at least the head, the importance of the head, because of the fossilized skeletons of monkeys. Certainly the majority reveal a bashed-in side of the head, so that the Australopithecine, the human or the pre-human, knew about the importance of the head because they could aim their clubs at the head of the monkeys with, of course, the desired result. But beyond that, the importance of the brain was certainly, likened to the technological marvels of the age. I'm going to go through the similes that have been used to describe the brain, but before that, I would like to mention that the ancient Egyptians didn't quite agree with the importance of the brain. They thought it was there merely to lubricate the nose. So what they did when embalming, they scooped out the brain matter from the nose. They went in with the scoops and scooped it all out and discarded it. They believed that the center of the organ responsible for all aspects of behavior, like the feelings and intelligence and creativity, was the heart, which was, of course, preserved separately in urns. Sometimes, quite a long time later, the technological marvel of the age was, of course, the Roman aqueducts. Those are marvels of engineering. And that nomenclature, the aqueduct, is still with us in some parts of the brain. You see, inside the brain and the spinal cord, we have a fluid. It's called the cerebral spinal fluid. Here, we have illustrated the various ventricles in which the cerebral spinal fluid resides. And with us, we still have the cerebral aqueduct, derived from the engineering marvel of the Roman Empire that connects the third and fourth ventricle in the brain. Now, the importance of the aqueduct, I'm talking about now the 13th to the 16th century, it was believed that the functioning of the brain depended on the ventricles. So that the ventricles were the seat of all the important functions of the brain. The actual brain matter that surrounded the ventricles was regarded as irrelevant. So we have Albertus Magnus, the bishop of Cologne, who had, he was the most prominent perhaps at the age, but a theory of ventricles. You see, they had only 3 there, and they ascribed all sorts of properties to these ventricles to explain behavior. And that lasted for several centuries until Andreas Vesalius. Now, Vesalius was a professor of anatomy at the University of Padua, near Venice. Padua in those days, in the 16th, 17th century, was the hub of the Italian scientific renaissance. Galileo was in Padua, and just next to Padua in Bologna is where the first university in the world was established in the 1100s. Now, Vesalius was a very straight and simple thinking man. He dissected not only human brains, but also the brains of other mammals, like oxen and sheep. And he realized that those brains also had ventricles, but they didn't have the reasoning powers of humans. So he decided that no, it isn't the ventricles that determine the intellectual functioning of an individual. It has to be something else. And coming right down the ages, when we come to the turn of the 20th century, Freud had another analogy for the brain, the boiler. It was a steam boiler. The id, I think it was, that was compressed, all the base tendencies and the drives that the humans have accumulated in the id, the id, ego, and the superego, and they caused pressure, and it was like a steam engine. That was so that the brain worked like a steam engine. A more apt analogy was that of a telephone switchboard. Now, the younger members of the audience probably haven't got the foggiest what that is. See, there were no, this is 1920s and 30s and perhaps even 40s. There were no satellites and no cell phones. So if you wanted to place a call, you lifted the receiver and dialed the number, and then a voice came and said, where should I connect you? And there were all of these switchboard operators that pulled out plugs and put in plugs and connected you to whoever you wanted to talk to. And they said the brain in those days was likened to a telephone switchboard, quite openly. It's in textbooks. So the brain operates like a telephone switchboard because you have an input through the eyes, the ears, the skin sensors, and then they go to the brain, and then the brain connects these inputs with the motor outputs, with whatever you want to do, with walking or reaching or stalking or whatever. So that analogy was certainly rampant. I did first year psychology in 1955, and they were still talking about the telephone switchboard, the way in which the brain operated. We'll revisit it in a little while. A more recent analogy is that of a computer. Now, that is almost compelling. Because the brain is like a computer. Now, you see, this is the current technological marvel. Like the telephone was a technological marvel early last century, computers are the technological marvel of now. So the brain works like a computer. Well, it doesn't. Nothing like the computer. I can expand on that later on, perhaps. But the point is that we will see how the wiring of the brain differs fundamentally from the linear progression of the information within a computer. Now, let's begin with the current state of affairs in what we know about the brain. We've gone through all of this historical stuff, and, the analogies, the metaphors that have been used. But now, well, what is illustrated here, by the way, is the initial part of the visual system. The eye, then it goes to the visual projection area, the primary visual cortex. In between, there is the lateral geniculate nucleus. And what happens is that when stimuli enter the eye, are first encoded in the retina. Now, beyond the retina, there is no light. It's all dark in there. You must realize that. The light stops at the retina, and that's not where we see. Beyond the retina, all we have is electrochemical impulses. And those impulses are coded then initially at the retina, and then they go from there to the lateral geniculate nucleus, where they get recoded. And at every station, including every part of the brain that we will see, that pattern of stimulation is recoded in new ways, in a way that is totally transparent to us, thank goodness. Now, what is the principal function of the brain? You see, the brain of a newborn can be considered a tabula rasa, a blank screen. on which experience makes its marks. Newborns have far more neurons in the visual brain than an adult. That vast number of neurons following experience, coalesce into local circuits called by by Donald Hebb, a great Canadian psychologist, the founder of the field of neuroscience. He referred to those as cell assemblies. They are neurons that connect with each other. Because when a certain, take a vertical line, just to be simple, that vertical line produces a certain pattern of activity in the visual cortex. Now, that pattern of activity is across several neurons. And those neurons that are activated produce synaptic knobs. They produce some growth. The principle is known as neurobiotaxis. So they connect. And when that line is shown again, the tendency is for the same neurons to respond until a cell assembly is established, which resonates whenever that stimulus is presented. Now, this is the simpler version, but what it is with experience, we establish neural circuits in the brain that resonate, that represent, that are turned on whenever we see that particular object. But we're not limited to object. You see, what is stored is action sequences. Or sequences, if you think of a song, "O Canada." Now, the sequence of the words and the sounds of "O Canada" are part of a group of cell assemblies that Hebb called phase sequences that become active. And the fact that they are active, then they generate a perception. Not quite, I will show you later how perceptions may, in fact, emerge from stimulation, but they certainly involve the activation of these cell assemblies and phase sequences that are established through experience very early in life. The process is mostly completed by the age of 2 or so, although it continues throughout life. Except that as you get older, the synaptic knobs are harder to establish. You know, when you're a kid, a simple instance will establish a cell assembly. Or when you learning a motor sequence, they do it much more easily. I learned to type when I was 50, and it was a hell of a job. And I still am not very good at it. I think I mentioned this, if you'll pardon me for dropping a name, but I was having dinner with David Hubel, and he said, he told me to, that was at the time I was learning to type. And he encouraged me to be persistent. He said, I mean, it's harder now, but those connections do occur if you persist. Instead of taking half a dozen tries, it might take you 50. But finally, the cell assembly will become established, and it did. But it's very nice. I don't know. But now, in fact, I wouldn't like to start to learn a new language like, I don't know, Urdu or something. It's very, very hard. But when you're young, things are so easy because these connections occur very easily. So we wind up with the brain being a vast repository of memories. But what we have stored in there is a representation of the world in all its actions, in all its chiaroscuros. It's our not only the physical world, but also the world of literature, the world of music, the world of sentiments. It's all stored in there in these phase sequences. And what's more amazing even perhaps is that we can actually tap into them. Here is the case of Dr. Penfield. Again, Montreal was really a hotbed of excellent research in this field. Not only Donald Hebb was at McGill, and Dr. Penfield was at the Montreal Neurological Institute. And there's people like Brenda Milner. It's really a good place to be. Also for the food, it's pretty good. So Dr. Penfield was a neurosurgeon. And one of the surgeries he performed was for the relief of epileptic seizures. That was done by cutting connections between different parts of the brain, mainly the corpus callosum. But before doing that, he had to make sure that he was not injuring vital parts of the brain, like having to do with speech or with motion. So what had to be done was to stimulate. The patient was awake. Okay, with local anesthetic, the scalp was open. The skull was opened, and Dr. Penfield could probe the nature of the processing being done in different parts of the brain with an electrical probe, a very weak voltage, but a very small tip. So he could, for example, stimulate parts of the inferior temporal lobe. When he did that, the patient reported in great detail hearing, one patient, a woman, reported hearing a symphony. And repeated stimulation of that particular spot caused perception of that specific symphony. And she could not be convinced after the operation that there wasn't a tape recorder hidden in the operating theater. She was the perception was so vivid that she actually heard the symphony when so stimulated. Another patient reported a visual perception, a downtown scene in downtown Montreal, reported the neon signs, the cars being driving past, and in great detail. So those memories are there. So we have this repertoire of memories, so auditory, visual, and emotional, that can be activated by stimulating directly the relevant part of the brain. Now, we see, not with our eyes, although the eyes are obviously essential, they are the port of entry, but we see with our brain. And one good example of that is the behavior or the perception of patients after cataract operation, but patients who were born with congenital cataract. Now, what is a cataract? A cataract, here is a section of the eye. This is the lens. The lens is just behind the pupil. And it is shaped like an onion. We've got layers. And as we age, those onion sheets become opaque, or spots die, and because there is no circulation in the lens, it is all the feeding and disposal of waste product is done by osmosis, which is a very inefficient way of getting things in and out. So it dies. And these days, one can simply go to a surgeon and say, look, you know, I need that, and if he agrees, he will do it in 10 minutes. I had my right eye done 3 weeks ago. Nothing to it. I mean, it took him 10 minutes. I had to go to a hospital, but I could have been sitting in a chair and he would have done it. Yeah, what you do is take out the old lens, or freeze it, shatter it, suck it out, and slip in a new one, a plastic, and that's fine. But that was not the case 60 or 70 years ago. But of course, you know, and now I can see a lot better, of course. But there are some unfortunate people who are born with congenital cataracts. So this is then the opaque lens they were born with. They can usually tell light from dark, but no shape, no color. And in the 1950s was when the cataract surgery was first introduced. And some people had the surgery done. One such case has been described by Oliver Sacks in The Man Who Mistook His Wife for a Hat, that book. So what happened? These people grew up to adulthood not being able to see. And all of a sudden, that opaque lens was removed, and they could see. But could they see? The point is, they couldn't. They could certainly tell colors, not by name, but this was different from that. They could follow the shape or contour of figure. In other words, there was input, visual input, but they could not identify what they were seeing. They were functionally blind. Even though light went in and went to their brains, you see, that repository of memories, that set of cell assemblies and phrase sequences had not been established for them. Certainly not with respect to vision. They had it with respect to audition and other senses, but not with respect to vision. So, it took 6 months for them to be able to tell visually the difference between a triangular shape and a round shape of wood. And it took forever to be able to, and never really well, to distinguish different faces. It was a problem for them to step onto the road and the traffic. Motion perception was virtually impossible. And in fact, most of those people reverted to wearing dark glasses and a white stick, and they carried on as though they were blind. And there were some consequences that were tragic, committed suicide because they went into a deep depression. So what we see with our brains. The light stimulus is merely a trigger to begin a sequence of operations, and we will go through them. Is someone telling me about the time? Because I have a tendency to go on and on, you see. Let me put my watch here. Oh, yes. What time is it? Yeah, well. Okay. Start yawning if you're bored. Then I will know. Where was it? Oh, yes, yes. With this post-operative behavior, we can certainly establish quite clearly that we don't see with our eyes, we see with our brains. And we hear with our brains, and we do everything with our brains. Now, oh yes, there is also another set of cases known as the agnosias, visual agnosias, in which parts of the brain, the visual brain, become inoperative because of stroke or injury. And in some cases, in visual agnosia, one can see an object like a cup, and can describe it, say the color, the shape, and the consistency, but they don't know what it is for. It's a case of visual agnosia. Again, even though we have a pretty excellent perception of an object, that is not all there is to it, because we must also know what it is for and under what circumstances it is used. And injury in certain parts of the brain causes impairment in that function. Now, yes, over the past few decades, we have learned much about the structure of the brain, mainly with brain imaging techniques such as MRI, CT, PET, and so on. We have now a pretty good idea of what is where in the brain. This is just a diagram by Fellerman and Van Essen. It's said to be the most cited graph in psychology, in showing you, that's a visual brain only, showing the connections between different parts of the brain. We will see how some of those connections are one way, almost all of them are two ways. Now, so we have then a pretty good idea of the structure of the brain, of the parts of the brain. But what are those structures? But how do they work? So we have a part list. What we need now is an operating manual. And that we haven't got. Not by a long shot, not yet. So we have a road, we have a map of the brain, but we beginning to know what is connected with what through techniques that highlight the pathways that connect different parts of the brain, DTI. What we don't know is how the functioning of those parts in connection with the other parts result in perception. The first step is to realize, and we're talking about current stuff now. We're past the Egyptians, and by a long shot. The first thing we must realize is that the brain regions that are identifiable because of cytological regions, shape of the cells and locations and so on. The various brain regions talk to each other with electrochemical impulses along two-way pathways. If area A sends a signal to area B, then it is almost invariably the case that area B sends a signal back to area A. Fine. One thing that puzzled people and continues to puzzle them, or some of them anyway, is that the descending fibers outnumber the ascending ones by a large factor. Take, I will show you in a moment, the connection, even at the very early part of vision, the connection between LGN, lateral geniculate nucleus, which is on the way to the brain, and primary visual cortex. Now, they're connected by a structure known as the optic radiation. Along the optic radiation that connects those two areas, the number of fibers that go up to the cortex are outnumbered by the fibers that go back down to LGN by a factor of 10. So for every fiber that goes up, there are 10 that come down. Why is that? It was the case until very recent, and even now, I get that sometimes when I give talks. Some of the colleagues say, oh, you know, that perception occurs because of a feed forward. Now, stimulus enters the eye, it is encoded here, encoded there, and then finally it gets to a higher level where perception occurs. And it is only a feed forward effect. So my question is, so what are all those reentrant feedback fibers doing? And in one case, at least, and I blush to say there was a colleague who said it, said, oh, that must have been an evolutionary mistake. So what do you say to that? In fact, what we have is a continuous forward and back and forward and back. So light enters the eye, goes to the back of the brain, to the occipital cortex, and then spreads to various centers, and those centers send signals back to the primary visual cortex. And we're going to see to have some ideas as to what happens when they go back there. But first I want to illustrate something that I did with one of my colleagues at SFU, John McDonald, in his lab with EEG. We had I think that was me, that had a cap, and he showed me a simple stimulus, and we were interested in finding out what happened, what kind of brain activity was triggered by a simple stimulus, like a flash on a screen. Now, what I'm going to show you is slowed down by a factor of 200. So it's 200 times slower. In fact, the actual thing is 200 times faster. And what I want you to notice is that there is virtually no way in which activation is only feed forward. It is both ways. It goes in, then it goes back, and it goes back, and have a look. Here's the stimulus. Back and forward. And that is the way in which the brain responds to a simple stimulus. It is all of this back and forth, which has been ignored by many of the people or researchers interested in finding out how the brain works and how perceptions arise. Let me examine a bit further this iterative processing. I will give you an illustration of what might happen, what probably happens. In fact, no, what we know happens, because I'm going to show you an experiment that illustrates that. So we have a stimulus, and that stimulus goes into the eye and goes into the primary visual cortex, where it is encoded in a different way. Now it's a different code now. And from there, it proceeds to other parts of the brain where it is encoded again. And from there, it goes back. Oh yeah, while it is there, there's new stimuli that come into the system, and they are there now. And now the reentrant stimulus performs a comparison. It is a correlation. And we do have in V1, in primary visual cortex, the circuitry that does perform a correlation. It's between, see, there are 6 cortical layers, and it's between layers 3 and 4. And the output then is, no, is it? I forget now. Sorry. The correlation is between 3 and 5, and the output is 4. But there is the circuitry there to perform a correlation between something that comes down from up top along layer 1 and something that comes from the environment in layer 4. But the correlation actually is between 3 and 5. And when that correlation is high, namely when the reentrant stimulus matches the stimulus that is there from the environment, then we have a perception. And when there is a match. If there is a mismatch, then nothing happens. A new hypothesis or a new sequence of events is generated. That is a reentrant processing. What does reentrant processing do? It does a number of things. And this is very current. Papers are being published now on this very issue. In one case, they enhance the perception of motion. Let me show you how. Here we have, in schematic form, the eyes, this is the lateral geniculate nucleus, LGN, the optic radiation, and visual cortex. Here is a slice, a horizontal section of a human brain. Here is the LGN. The optic radiation is here, that connects to the primary visual cortex. Now, it is this communication between these two areas that I'm going to talk about how the reentrant processing enhances the perception of motion. The reentrant processing meaning the communication between primary visual cortex and LGN, V1 and LGN, how that iterative communication enhances the perception of motion. That is done, by the way, with a technique known as microelectrode recording. Let's say this is the brain of a cat, and we can monitor the activity of a single neuron in the cat's primary visual cortex, which is area 17. In us, it is called V1. It is equivalent. Well, not quite, but similar. And this is a microelectrode. It is a microelectrode. The tip is so small that can rest against the wall of a neuron and monitor the electrical activity of that neuron. So that when a stimulus is presented, then the activity is recorded from that microelectrode. That's how we do it. Now, so here we have then the section of the brain, and we're going to monitor the activity, the reentrant activity along the optic radiation. I'm going to now have a schematic representation of LGN. Here are all the cells in the lateral geniculate nucleus, schematically, in the dorsal LGN, by the way. And here is area 17, or primary visual cortex. And we're going to see how communication between these two areas along the optic radiation enhances the perception of motion. Let's have a stimulus. Fine. It is recorded in LGN. And that unit in LGN sends a signal up to primary visual cortex. Fine. We have no motion yet. But we make this move. In that case, then we have, it occupies, it generates activity in, it activates another neuron next to the previous one and sends a signal back to primary visual. Now, in primary visual cortex, we have, and cats have, and monkeys have, a unit called Reichardt motion sensors. They require two inputs in sequence. And then when the timing between the two inputs is correct, they generate an output, and that is a motion signal to the brain. Of course, we don't see motion. There's no motion in the brain. There's only coding, and motion is coded in terms of these Reichardt motion sensors. And now we have two sequential stimuli in primary visual cortex, and now the Reichardt sensor in primary visual cortex knows what motion the direction it is and sends a signal back. To LGN. Why? That green signal causes the threshold of the unit just ahead of the moving stimulus to be lowered, namely to be more sensitive, so that the sensitivity of the neurons that are in the expected direction of motion becomes higher. And so we can perceive motion more clearly. Not only that, but there are signals, by the way, that, and I haven't shown it here, that go down back towards the units that have been activated just before to suppress it. And that is to suppress motion smear. And all of this is hardwired. We're born with it. And this connectivity, you see, that's why we need 500 trillion connections. The Reichardt sensors are oriented everywhere. We have as many as we need. And that is why we know that the descending fibers from cortex to LGN far outnumber the ascending one, because they have to predict, they have to cover all expectations. Yes. Yes, I will be faster. That's one way of checking on reentrant processes. Another way of checking on reentrant processes is by TMS, by transcranial magnetic stimulation. What is it? Well, we have a coil here that generates a magnetic field like this. And when that field is generated, then it interferes with the electrical activity of that particular spot in the brain, about 1 cubic centimeter. Even better if we have dual cone, a different kind. But it interferes with it so that it doesn't operate, so that when it is zapped, that area momentarily, not momentarily, briefly loses its function. Now, we can use TMS to disrupt activity at different stages of processing. Let me illustrate. Here is a stimulus. Now, we can present TMS before the stimulus enters the system. Or we can present TMS once the stimulus is in the primary visual cortex. In that case, it will interfere with the perception of that stimulus. Or we can present TMS while the stimulus is upstairs somewhere. And in that case, there will be no interference. Or we can interfere with the stimulus when it is re-entered, and in that case, it will interfere again. So we'll have 2 times in the sequence of events with which we can interfere with the stimulus. Now, here is an experiment that illustrates the sequence of events very nicely. It was done by Courtauld et al. In 1999. Now, we can try the first option of presenting, this is a time scale, by the way, and it's in milliseconds. Now, 1 millisecond is a 1,000th part of a second. We can, in this case, we will deliver the TMS pulse here before we present the stimulus. So we present the TMS here, and now we present the stimulus, and it goes in there, and it is encoded. It goes on to be recoded, and it goes back, and it generates a perception. So presenting the TMS pulse before the stimulus has no effect, provided it is long enough before the stimulus. Now we're going to try the other pattern, of course, and we have performance. This is how accurately the stimulus, the subjects identified the stimulus. In this case, they identified it correctly 80% of the time. So perception was not impaired. Now we'll consider the case in which the TMS occurs after the stimulus is, of course, we can present the stimulus 10 milliseconds after, 20 or 30 or 40. Here it is 20 milliseconds after the stimulus is presented. Here is the stimulus, goes in. Now we zap it right here. And nothing happens. That is, the stimulus is totally wiped out, and performance is now down to 45%. Correct. Now we'll try the case in which we wait until the stimulus is upstairs. And so we present the stimulus, and then we zap it at 60 milliseconds. Here's the stimulus, goes in there, it is encoded, it moves up there, and now we zap V1. Nothing happened because it goes back there, and it perceives the stimulus the subject quite nicely, and performance is very high again. The next step is to move the separation between stimulus and zapping even more, but now we're going to zap it when the stimulus has returned to has re-entered the visual cortex. First time round, encoded, encoded again, go back, and now we zap it. And by Jove, perception is totally impaired, and performance is now quite abysmal, at 30%. And finally, we can present the stimulus, the TMS pulse after the entire cycle has been completed. And of course, in that case, now we have perception, and we present the TMS, and it has no effect. So you can see that with TMS, we can follow the stimulus inside the brain as it is processed. Confirming what we know from neuroanatomical sources, that the working of the brain relies on reentry. It's not merely a feed-forward sequence of events that begins with stimulation in the eye and culminates with the perception of an object or whatever. We have this continuous communication between centers of the brain, and that's the current knowledge of how the brain works. I'm going to shortchange you, because I had other material I wanted to show, but I won't. And I will end by referring to the common belief that we use only about 10% of our brain power. Well, is it true or false? There has been a current movie, Lucy, with Morgan Freeman and Scarlett Johansson, about a scientist who discovers how to activate that 90% of the brain. Don't you believe it? We use the brain, all of the brain, all of the time. It is active all of the time. When we sleep, we dream. When we're awake, we also dream. Sometimes. But there it is. This is how we now believe that the brain works. I don't know whether you have to turn it on.
I think it's working. Anyone with a question for Dr. Lollo? I've got one for you, which is that when you're talking about the reentrant processes, going back down to LGN, it reminds me of the schematic diagram for tuning systems that we talk about in courses that I've taken in the past, at least. To what extent do you think some of the reentrant processes are in fact tuning mechanisms for lower systems?
Could you? I didn't quite catch the point. Did it remind you of what diagram?
So when you're talking about the projections from visual cortex back to LGN,
yes.
The reentrant, that reminds me of diagrams for tuning circuitry.
Tuning circuitry.
Tuning circuitry. And
Yes,
to what extent do you think reentrant processes are in fact tuning systems?
They are. Yes. You see, because they are a feedback circuit. Well, first of all, some of the reentrant signals go to the very cells that initially generated the activation. And how can I put it differently? In fact, I can answer your question best by pointing to how the perception of motion is enhanced by reentry. That itself is a tuning, namely tuning the system to motion in a certain direction. But I have a sense that that is not what you meant by tuning. What could you tell?
I was feeling more of the distinction between sharpening the tuning of an individual unit versus lowering its threshold.
Yes, of course. Yes. The reentrant signals are not only excitatory but also inhibitory. So, speaking loosely, they can serve to narrow the focus of attention, if I can use that word, and that is tune the system to a particular shape or particular spatial frequency. And yes, there is no other way that I can think of in which tuning can occur other than by reentry.
Yeah, one of the comments that Maria made when we were talking about this a little bit was that the concept of a reentrant process going back and forth, that oscillation that you showed with recordings of your own brain, it seemed, wasn't a familiar topic, not in the not so distant past in neuroscience courses. There was some discussion of a tuning system, maybe some feedback. I was just curious in watching that if there is a sense of how many oscillations or reentrant back and forth processes can occur in some systems.
Yes. One complete cycle between V1 and LGN, complete all the way down and up, can take as little as 5 milliseconds, but it is between 5 and 10. Many of those cycles can occur in a matter of 100 milliseconds. Not only that, but you see, these reentrant circuits, these loops, occur throughout the visual system. The longest is between prefrontal cortex and V1. Now, everybody talks to V1. All parts of the brain are connected directly or mostly directly to V1. So one cycle between V1 and prefrontal, in fact, even more, inferotemporal cortex, in the monkey takes about 120 milliseconds. So we have all of this brain activity that occurs concurrently, very small loops, local loops with long loops that take longer. It is, they're all in the sense, if you, by tuning, you mean increasing the adaptability or the sensitivity or narrowing the scope of perception, then they're all addressed at that purpose.
Thanks. Any other questions? All right, well, looking at the clock, Ellen, what room are we meeting in?
We're going out this door and just to the left.
Out and to the left, come and have a glass of wine and talk to Vince off camera.